Hi everyone, welcome to the Wise Tutor YouTube channel. Today we are bringing you video number 7 of the AP Biology sequence, Unit 1 Biochemistry, and this video is about dehydration, synthesis, and hydrolysis. So when we're talking about these two processes, we have to remember that right before this, we were talking about the four macromolecules and how each one has its monomer or building block and builds up to a polymer or multiple units combined together or bonded together with a specific linkage or bond. And so these processes are the two processes that build up or break down those macromolecules. Okay, so let's take a look. I think this table really summarizes the information well between the two. Dehydration synthesis is also called condensation, and in this process we go from a monomer to a polymer. So we are making the substance or the molecule bigger. Okay, we are going from monomer to polymer, and this is called an anabolic reaction because it is becoming larger in size because you're adding more and more monomers to it. Okay, andergonic. This is an andergonic reaction because it requires energy, and basically it's characterized by the removal of water molecules from these organic molecules. So as we keep building up or keep adding on more and more monomers to a chain of polymers, and we're bonding those together, we're also removing water in between those monomers. We're getting rid of those water molecules. They come out as products, and so you'll see that on the next slide. Hydrolysis literally stands for break by water. Hydro is water, and lysis is break. We'll actually discover lysis a bit more, or like talk about it in the cells unit. Anyway, this one is the exact opposite as the condensation reaction. We are going from a larger substance, a polymer, to a monomer. All right, and it's called a catabolic reaction because it's getting smaller or reducing complexity. Pretty much, you could think about it. It is an exergonic reaction. It happens spontaneously and doesn't require energy. Okay, and you add water molecules instead of removing them to these organic molecules so that they can break the bonds between these monomers so that we could result in a monomer and not a polymer. So let's take a look in each macromolecule. What do these processes look like? Okay, here is a carbohydrate. You've got two monomers, two glucose monomers, and you're adding them together. Notice the red highlights. This is exactly where the H2O or water molecule comes from. It comes from an OH hydroxyl group on one monomer and the hydrogen from the hydroxyl group in the other. What actually ends up happening in this is that you form this glycosidic linkage with the remaining oxygen. This one is this one, and you form this glycosidic linkage and you form one molecule of water. All right, so the red highlight traces the water all throughout the reaction. This is which out of the two reactions that we were just talking about? It is dehydration synthesis. We are making synthesis, making a larger compound. We are now looking at a disaccharide instead of instead of two separate monosaccharides. So here is the disaccharide. Now the opposite is also true. You could start off with a disaccharide with the glycosidic linkage here, and you could add water to it, and the opposite thing will happen. This glycosidic linkage will break, right? And instead, we're going to use the water, we're going to break the water and use it to reform the two individual monomers. All right. In proteins now, dehydration synthesis. In proteins, you've got the NCC. That's protein characterization there. And the same thing happens. You're gonna form hydrogen as you combine the two molecules to get a dipeptide. Okay, and you see the peptide bond forming right in between. Hydrolysis is, of course, the other way. We're going to start from the dipeptide, add water to the molecule and to this bond. The bond breaks, and we end up reverting back to the original structure where there's a hydrogen attached to this nitrogen, and to this carbon we have a hydroxyl group attached. All right. In lipids now, lipids, as we said, we have the glycerol backbone, that is the structure, and a fatty acid. All right. And we have the hydroxyl group right here and the hydroxyl group on the fatty acid. So the hydroxyl group from the fatty acid is actually the one that's fully removed, and we remove a hydrogen from the glycerol group or backbone. All right. This results in the formation of a H2O molecule and the formation of an ester linkage between this oxygen and this carbon, because we've gotten rid of all the molecules in between those two, all the atoms in between those two. And so that results in an ester linkage forming. Now, since the glycerol has kind of three places where the fatty acids could happen, or like bond, a lot of times you'll see this process with 3H2O. That means for every one of these fatty acids, you're removing an H2O from the system. Now, the opposite is true for hydrolysis. Okay, hydrolysis, you're putting in the water to the system, and instead of having a polymer with a glycerol backbone attached by ester linkages to the fatty acids, you're trying to break those ester linkages apart by adding water into the system in the same locations that you were removing them in dehydration synthesis. So I'm pretty sure this is like you're starting to get this. And now we're going to look at nucleic acids, because this one is kind of not really explained often in AP biology. It's kind of one that you might not really need to know or pay attention to compared to the others. The others, I would say, are pretty important and you need to know them. But we could go through this. So this is, it could be a DNA, or in this case, it is a DNA strand, because it's including thymine instead of uracil. But this could really be a DNA or an RNA strand. And you've seen in our previous nucleic acids video the structure of a single nucleic acid with the phosphate group in red here. You've got the ribose or deoxyribose pentose sugar pretty much, and you've got one of the four base pairs or nitrogenous bases, right? There's purines and pyrimidines. We all go over that in the nucleic acids video, so if you want to check that out. But anyway, you see right here the formation of the phosphodiester linkage, and this is how you link nucleotides together to form a polymer of them. And so what happens is this. On here, the H here is attached to an oxygen right here as well. So this OH group combines with the phosphate's hydrogen, and what ends up forming is this phosphate group and this phosphodiester linkage. And if you look closely, you'll see that still, in order to make a polymer, you are ending up removing the H2O molecules each time you add two nucleotides together. The opposite is true if you want to break down this strand of DNA or RNA. You would insert H2O back into those initial spots. All right, so you'd insert an H here and the OH on the other end of the ribose sugar, or the deoxyribose sugar. All right, and that is it for dehydration synthesis and hydrolysis. And I hope you guys enjoyed the video.